Next up, we have Dr. Barbara Bekins. Dr. Bekins is a research hydrologist for the U.S. Geological Survey Water Mission Area located in Menlo Park, California. She has studied the fate and transport of crude oil contaminants in groundwater for over 25 years. She serves as research coordinator for the USGS crude oil spill study site near Bemidji, Minnesota, for those Minnesotans, that's up there in Lake Country, and is a member of the National Academy of Engineering. As something fun, during COVID-19, she has been enjoying finding recipes for unfamiliar produce such as fava beans, which are delivered weekly to her in a CSA farm box. So thank you so much, Dr. Bekins, for joining us today, and we look forward to your talk.
Thanks for having me. I'm very happy to be able to present our results for a terrestrial spill. This is a terrestrial oil spill site. It's been studied by the USGS and many academic partners that we've worked with over the years since 1983, so 4 years after it started. The site today, management of research at the site has always been funded by the USGS, but in the last 8 years, it's also been partially funded by Enbridge Energy, who's the responsible party at the site. They acquired the Lakehead pipeline system where the spill originally occurred in 1979. So the site has a rich data record and infrastructure to continue to aid our understanding of the long-term fate of oil in the environment. And this, my title slide here is an overview of the spill at the time it occurred, with all the areas impacted by the oil outlined in the dashed yellow line, and a full-size tanker truck is there as a white square, so you get an idea of how much was impacted. The break was here along the pipeline, and it was under pressure, so the oil sprayed out over a large area, and then it also flowed along the ground. Okay, so you should be seeing my second slide soon. So it was line 3 that broke, and actually line 3 has been responsible for the 2 biggest terrestrial oil spills in the US. And line 3 is a line that Enbridge is trying to work with Minnesota and the tribes of Minnesota to replace in the future. So as I said, it was a buried line under 500 PSI pressure, 34 inches in diameter. So the spill was a little bit larger than the one we just heard about in Alabama, 441,000 gallons. It was a light crude, and the cause was a bad weld on the pipeline. So line 3 has kind of been a bad actor for a while. Here's just a schematic of the bad weld. So the site's located up here outside of the town of Bemidji, near the source of the Mississippi River. But here's another aerial photo of the site at the time of the spill. There used to be railroad tracks here. This was the pipeline right of way. You can see that the trees are cleared for the pipelines. There were just 3 pipelines now there's 6. So this was the area that was sprayed. So it was forested, and the trees were coated with oil. And then that sprayed area, the oil kind of flowed along low areas to a wetland. Okay, so my third slide, I'm showing you a series of photos illustrating the emergency response at the time of the spill. So the emergency response was estimated to have removed about 75% of the spilled oil. Most of that by pumping, 83% by pumping. And then excavation and land farming, so they dug up a lot of contaminated soil. That accounts for 6%. And then finally burning or evaporation, so you can see a picture here of the burned oily vegetation that was used to remove about 10%. So the excavation, this site is a glacial outwash sand and gravel aquifer from the big continental glaciation. So the oil soaked in within days into the aquifer, and now it's at the water table. So here's a Google Earth image of the site today. It is managed for logging. It's on state forest land. The pipeline right-of-way runs through here. The site of the pipeline break is here in the yellow star. The blue areas show where the oil moved along the surface. The yellow area is the spray zone. And notice how lightly vegetated the spray zone is. I'll show you another picture of that. The three black outlines are where the oil is sitting at the water table and providing a continuous source of contamination to the groundwater, which my talk will mainly be focused on. The white spots are mostly observation wells, and then we also have some locations along the shore of this down gradient lake. So groundwater flows along this line of wells, kind of left to right. And the plume, this down gradient lake is about 300 meters from the center of this oil body. So the spray zone I mentioned is very lightly vegetated, and the reason why is because the soils are hydrophobic. So the spray zone was never contaminated below the surface, only the top meter was affected, and no hydrocarbons and transformation products. The hydrocarbons and transformation products are transported to the water table, but not the oil. So this is a close-up of how water beads up on the surface. So this is the cycles of heating and cooling of the oil-coated sediments leads to this hydrophobic behavior. And here are some students from the school of St. Paul that you can see them playing with the hydrophobic soils. I'm going to spend the rest of the talk on the groundwater contaminant plume, since this is a seminar about water contamination. So here's an overview of the aquifer with the lake in the green, showing monitoring wells. In 1996, this was the contour of the 10 microgram per liter benzene concentration in the well, as mentioned, measured in the groundwater with the wells. And then coming forward to 2010, you still see just 3.3 micrograms per liter of benzene, so below the MCL at about the same location in the aquifer. So the natural biodegradation is doing a good job of controlling the benzene plume at this site. And the main control on the degradation, this is anaerobic because there's so much oil in the aquifer, but there's a large supply of iron oxide and iron oxyhydroxide coatings on the aquifer sediments that is coupled to anaerobic biodegradation at the site. So this was the site where anaerobic biodegradation coupled to iron reduction was first documented. So next I'm going to show you a cross-section along this line. So these are our wells, and you can see there are a lot more up gradient. This was outlined in red as the 1996 contour of the benzene. And what you can see is that, so the benzene plume is still being controlled in this area. We don't have a lot of wells down gradient, although we've been adding them because some parts of the plume are moving down gradient. And there was an attempt to pump more oil out of the aquifer between 1998 and 2003, and the pumped water was infiltrated here and formed a secondary plume under the main plume. So most of the plume mass today is non-volatile dissolved organic carbon. This shows a line graph of distance along the center line of the plume, concentration measured in the groundwater for non-volatile DOC, and also for benzene. So the benzene is way down here at about 3 milligrams per liter. In contrast, the DOC is almost 30, so an order of magnitude higher in 1995. And then by 2016, so about 20 years later, you're seeing that the plume has expanded by about 10 meters. So it's expanding about 2 meters a year. And the reason for the expansion is because there's a finite supply of iron oxide on the sediments, and that's consumed during the biodegradation reaction. So the plume, but the good news is there's a very robust decrease in concentration due to biodegradation where iron is present. But the biodegradation doesn't go to completion of this DOC. This is the background DOC. And so you can see that some of the DOC is reaching this lake down gradient. So I just want to, my talk is going to be very focused on present day methods for characterizing these sites. So a lot of you are probably familiar with the total petroleum hydrocarbon measurement in the diesel range. So that's generally required at oil spill sites. And it just is liquid extraction from the water with a GCFID analysis, and all compounds that elute between and the normal N-alkane C10 and the normal alkane C28 are counted regardless of whether they're hydrocarbons or not. One problem with the liquid extraction is that much more efficient extraction methods for water are used today for DOC. So here is a comparison published by Phoebe Zito and colleagues. Phoebe's at the University of New Orleans. It's showing how much carbon is recovered with the liquid extraction in black, and how much is covered with 3 different solid phase extraction methods, where the best one is the Oasis HLB extraction, which is going to be used for the rest of the results in the study. And here's the background site for comparison. You have the same extraction efficiency across, these are different distances in the groundwater contaminant plume, 39, 102, and 254. So because researchers noticed in 2003 that more than just hydrocarbons were coming out and being quantified in a total petroleum hydrocarbon method, they proposed that silica gel cleanup be used to remove natural organic matter and polar transformation products of the hydrocarbons. So this has been very controversial. Some local regulators have allowed it. At present, other state regulators are developing policies about whether transformation products should be removed from a total petroleum hydrocarbon analysis. So this, this controversy has huge implications for those of us who are concerned about water quality impacts of oil spills. So what I'm showing here is, again, data from different wells at different distances from the spilled oil. These are samples collected from the groundwater contaminant plume. The vertical axis is concentration. So the total DOC here goes between 22, dropping to about 2.5 milligrams per liter down gradient. If you do a total petroleum hydrocarbon in the diesel range, you see less than 1/3 of what the total that's there. And if silica gel cleanup is allowed to remove the polar compounds, you see only about 6% of what is there. And you get below detection for these last two wells in the plume. So our interest is in exploring what does that mean for the potential health impacts of evaluating this water for potential health impacts. So in 2018, we went to the field, we sampled 5 wells, 4 in the plume and 1 upgradient well. So 3 of the first 2 wells were in the hydrocarbon plume. The last 2 wells were in the plume, but there were only metabolites. So silica gel cleanup detected no true hydrocarbons at these last 2 wells. So we performed 2 types of screening. The first type was a bioeffects screening where human liver cells are used. So these are these human liver cells are transfected with 46 different transcription factors of one type and 48 of a different type. So these are transcription factors that are known to be associated with toxic response. The wells are, the cells are exposed to the evaluated compound, and the response of the cells is measured by the amount of transcripted DNA of lengths that are dummy lengths that vary with the transcription factor. So the dummy lengths can be measured by capillary gel electrophoresis and fluorescence to determine the degree of response of each transcription factor. So these are the first of the results from the site. We mainly got responses in 2 transcription factors. One was the aerohydrocarbon receptor. So the vertical axis on this plot is upregulation compared to just a solvent control. The horizontal axis is the concentration of non-volatile DOC in the plume. So these higher concentrations are from near the spill, and the lower concentrations are from down gradient. So what you see, and we also got upregulation of the pregnate xenobiotic receptor. What is really important about this is that silica gel cleanup, so the true hydrocarbons in the plume, we got a response that's the same as what the background site showed. So a 1x, no upregulation from the true hydrocarbons at any location in the plume and for either receptor. The liquid extracted portion of the chemicals, we did see a response. So this is what would be extracted for a typical total petroleum hydrocarbon measurement. And finally, if we use an extraction method, a solid phase that gets the most possible carbon, we see an even higher response of these transcription factors. So you're completely missing the toxicity if silica gel cleanup is allowed. You're partly seeing it if you're extracting what's seen in a total petroleum hydrocarbon method, and you're seeing even more if you do a more efficient extraction. So the second method that we used was a chemically activated luciferase expression. So again, on this axis is the non-volatile DOC concentration. And on this axis is the response of the luciferase in these human liver cells. So this is the background site. This is 1 mil water equivalent at 10 to the 0. So the mass that's added is the same as the mass in 1 mil of the water. So here at the background site, you see no response until you get to 40 times the in situ concentration. And then you see responses in the groundwater plume with the highest response at the closest well to the oil, 68 meters down gradient from the source. And then lower responses with distance, although still a high response at 254 meters. So these responses can be normalized to a known positive control, which is a beta-naphthaflavone, a PAH, that's typically used to test that these AHR responses are occurring in the test sample. So here is the response normalized to the beta-naphthaflavone with distance from the oil body. So the calyx response is shown here in red. So you get the highest response closest to the oil, with the response dropping with distance, but still a response that's equivalent to 100 nanograms per liter of the beta-naphthaflavone control. So you're still seeing a response that's associated with 100 nanograms of a PAH. And the response relates better to NVD, to the DOC, than it does to the TPHD. So in conclusion, the required analyses at hydrocarbon sites underestimates the polar transformation products that are present. The plume is mostly polar transformation products at this point in time. The polar transformation product plume is expanding because the iron is being used up on the aquifer sediments. And in vitro cell assays show that the transformation products should be assessed further for toxicity. And my last slide, I'm just showing an illustration of our future plans. We're planning to do in vivo response of a fish embryo. This shows zebrafish. We'll likely use fathead minnow embryos, would be the next step for our research on these compounds in the plume. Thank you for your attention, and I'll be available for questions on the panel.